Hi there. In this video I'm going to talk through epistasis or epistasis, depending on how you want to pronounce it. In the AQA specification they expect the use of fully labeled genetic diagrams to interpret and predict, and we've got epistasis specifically mentioned in the spec. Okay, so epistasis is where the expression of one gene is affected by the expression of a different gene. So an example can be hair color and baldness in human males. So if a human male can have blonde or red hair, but they've inherited the baldness gene, well, having yellow or blonde or red hair is irrelevant. So that's an example for humans, but specification tends to talk about fur color in rabbits and dogs and horses, etc. So we'll do mice. So we're going to look at the agouti coloring in mice. So for mice, they can have three colors. Agouti, which is a kind of a browny color, and agouti is dominant, and we're going to call that capital A. And they can be black, and that is going to be small a. They also have a gene for color, which we're going to say is dominant, capital C, and no color or albino is going to be little c. Okay, so I'm going to do a full cross here for F1 and F2. So let's just work through it. Parental phenotype, we're going to go with homozygous dominant and heterozygous dominant, and that's going to be for color, he's going to be big A, and big C, and homozygous recessive. I'm going to cross that with little a, and little c. So while we've got the black color there, since we've got the homozygous alleles for color, it's going to be, with homozygous recessive alleles, it's going to be albino. Okay, parental genotype. There they are. So we have the phenotype is, for this one is agouti, and this one is white. All right, let's do some alleles. Big A and big C, parental genotype, possible of big A and big C, and possibility here is 2 big A, big C, and little a, little c, little a, little c. So let's cross our male and our female, F1 generation. So let's say the male was big A, big C, and female was white. Okay. So what do we end up with? Big A, big C, little c, big A, big C, little c, and again. So as usual, we end up with 100% heterozygous. So offspring genotype, big A, big C, little c. The phenotypes, they're all agouti, 100%. Okay, so our ratio of phenotypes, 100%. All right, so let's do an intercross and with our F1 generation. So we'll start off with parental phenotype. We'll have agouti crossing with agouti. Genotype, heterozygous. Gametes, big A, big C, big A, little c, little a, big C, little a, little c, and that's the same for both. All right, so let's cross our male and female. So let's put male on the top, female down the side. Big A, big C, big A, little c, little a, big C, little a, little c, and down the side. All right, so this is looking like a bit of a dihybrid cross. Let's be careful. Big A, big A. I'm going to do my A's first. So all homozygous, and then we've got a hetero, big A, little a. Now do the color, big C, big C, big C, little c, big C, big C, big C, little c. And let's keep going. Big A, little a, big C, big C, big A, little a, little a, little a, little a, little a, big C, little c, big C, big C, big C, little c, big A, little a, big A, little a, little a, little a, little a, little a, big C, little c, little c, little c, big C, little c, little c, little c. Alright, that looks neat. So let's interpret what we've got. I'm not going to rewrite all of my F2 offspring genotypes because I've done them all here in the Punnett square. So let's look at the phenotypes. So I'm just going to use a different color here. Let's find all the colored and agouti mice. So colored and agouti, there's 1, 2, 3, 4, dominant, dominant, no color, dominant, dominant, agouti, no color, dominant and dominant, and another one, dominant but not agouti, dominant but not agouti, dominant and dominant, no color, color but not agouti, and no color. Okay, so we've got an agouti mice. We've got 1, 2, 3, 4, 5, 6, 7, 8, 9 of them. And what else we got? I'm going to use my purple pen to find my albino mice. So we need no color and recessive. So, well, just no color will do nicely. So that one, and there's another one, and there's another one. So our homozygous recessive, there's 1, 2, 3, 4 of those. So I'm going to put him over here. So 4 albino. And let's find the colored mice that are homozygous recessive for color. So you can see they're nicely grouped here. So I'm just going to cross them 1, 2, 3. So we have 3 black. So those ratios work out as 9 to 3 to 4 when dealing with epistasis. Now there's different types of epistasis. There's recessive epistasis and dominant epistasis. This is a recessive epistasis because the colored gene is homozygous recessive. But if you reverse its expression and redraw the Punnett square, your numbers will change and your ratios. Why don't you give it a go?